guys, I'm Pauline and welcome back for a new Pauline's macro video. So today I am keen to present you Emilie Boutet. She is another PhD student. She is actually working on the same floor than me because we belong to the same department in Armand-Frappier Institute. And so I know her well because she's currently training me on radioactivity because I want to mark a DNA to be able to run a jet shift experiment. So what I can tell you about Emilie is that she loves making sciences popular and accessible to people. We worked together on a project which was to make poster for children for middle school children, and like we have to really make sciences and our project or the lab project really easy for them to understand. And I know that she also writes a lot of articles about sciences in general. She recently wrote with a supervisor an article to make things clear about the coronavirus and the HIV. And I do remember that I watched a video of her explaining what was a virus and how, especially, the coronavirus is working in cells for everybody, not scientific people. So that's why I asked her to present to us her project in a popular way because I knew that she will accept it right away. So I will give you some information about her works in the description of the video. But for now, I let you enjoy her project and how she made bacteria working for her. So I hope that you will have a lovely listening.
Hi everyone! So today I'm going to talk to you about my project, which is called the Smallest Industrial Workers You Ever Hired. So first I'm going to introduce myself. My name is Emily. This is me on the picture. I'm a PhD student at INRS Centre Armand-Frappier Santé Biotechnologie. It's a small university in Montreal. So my academic background, I did my bachelor in science at McGill University with a major in biology. And then I started my graduate studies at INRS. So I did my master's in applied microbiology and I switched into PhD in biology. This is my PI, Jonathan Perrault. So we're quite a big lab. Our main research interest is everything related to non-coding RNA, more precisely bacterial non-coding RNA. So these RNA elements are really important in gene regulation and there's many more to be found. Another important aspect of our lab is that we develop tools based on nucleic acid to use them in different applications like biosensors, synthetic biology, or metabolic engineering. My project actually combines both aspects of the lab research interest, so I'm using non-coding RNA to develop a new biotechnological tool. So the key to mass production can actually be on a very small scale. Bacteria are already able to produce several compounds that are found in our everyday life, like proteins, vitamins, and antibiotics. So why not use bacteria as a small microbial factory? So the compound that we want to produce is succinic acid. It is found in everyday products like plastic, cosmetic, and dyes. It is normally synthesized from fossil fuels like petroleum, but there's actually an interest to have a more bio-based production of succinic acid. The cost of succinic acid is quite high because it's closely related to the cost of the raw materials used in its production, like fossil fuels. So there's a great interest to develop new tools to produce succinic acid, like using bacteria, for example. One bacteria that is really interesting for this matter is Methyllurobum extorquens. It is a pink pigmented bacteria. It's a great candidate to be a biotechnological tool for the production of succinic acid because it feeds on methanol. Methanol is a type of alcohol that is inexpensive to use and can be found in renewable sources like municipal waste or from byproducts of the chemical industry. So using MX as a microbial factory uh, actually kills two birds with one stone. First, we can use a waste products that cost nothing, methanol, to produce something that is value added, that is more expensive, succinic acid. Jackpot. Also, uh, it makes it possible to detach the production of succinic acid from those of non-renewable energy sources. So MX is naturally capable of producing succinic acid, but it feeds on the compound as soon as it has made it. So how do you stop it? The trick is to rewire MX circuits to prevent the bacteria from consuming succinic acid when it is produced. So as you can see on the picture here, this is the way MX can metabolize methanol. So within the TCA cycle, there's actually succinic acid. So if we play with the circuit, so if we modify the genes that are responsible to consume succinic acid and those that are responsible to produce succinic acid, we can actually allow succinic acid to accumulate within the growth medium. So the process by which scientists modify genetic material is called genetic engineering. So in our lab, with some collaboration with other labs, we were able to produce a new MX13 strain that is able to actually produce succinic acid. And there was a patent that was published on the subject. The challenge now is to find ways to optimize that circuit so that even more succinic acid is produced at a faster rate and a lowest cost possibly. So this is where I come in. So my goal is to develop genetic tools to improve succinic acid production using genetic engineering. So I already have the strain that is able to produce succinic acid, but my goal is to improve it. So yeah, that's it. That's my project. I hope you liked it, and thank you for listening.